So we've seen a portion of this video before. We've watched the first minute and a half already in a previous video, and we've paused it at that time, and I said, okay, well, we're going to stop here because this continues on the discussion of replication at least, and that's something that we hadn't talked about, but now we have. So we kind of get this idea of what replication is. And we've talked about DNA basically being separated into the two strands by using something called a helicase. And these two strands are beginning to get copied. And one continuously gets copied in one direction without a problem. The other strand, though, has to get copied into fragments, and those fragments basically have to be glued back together with another protein called the ligase that just makes sure everything is smooth and consistent. So this video is going to kind of show you a visual representation of what goes on with DNA replication.
Using computer animation based on molecular research, we are now able to see how DNA is actually copied in living cells.
Okay, so at this portion of the video, what you're seeing is the helicase. I mean, this is the helicase. This is kind of the four-finger protein that we saw a while ago. And this end is the DNA parent strand. So the DNA is getting fed into the helicase, which is here. And this DNA is basically getting ripped apart. And you'll see that in the video. It gets ripped apart into two strands. One strand is fed over to this finger and beginning to get copied. And that's why you see another coil that happens over here to the right hand side. The other strand is getting fed to this finger back here in the back, and this is just a single template strand right now. That's all that is, just the parent strand. And this is getting copied into the fragments. So this is the Okazaki fragments, and you're going to see that. It's going to basically start creating a loop in the video. So one of these is continuously synthesized. The other one is getting synthesized in fragments called Okazaki fragments, and you will see what the protein does in order to just keep everything streamlined and to prevent this thing from turning into a huge, horrible knot. I'll hit the play button and you can kind of follow through and listen to her.
You are looking at an assembly line of amazing miniature biochemical machines that are pulling apart the DNA double helix and cranking out a copy of each strand. The DNA to be copied enters the production line from bottom left. The whirling blue molecular machine is called helicase. It spins the DNA as fast as a jet engine as it unwinds the double helix into two strands. One strand is copied continuously and can be seen spooling off to the right. Things are not so simple for the other strand because it must be copied backwards. It is drawn out repeatedly in loops and
so this is the strand that's getting replicated continuously. So the DNA strand is coming over here. This is the parent strand going into the helicase. The helicase separates it into two strands. One strand is being fed here, and that's getting copied as it goes. The other strand is getting fed here in this loop, and this is the fragment. So what's happening is that this fragment's getting spit out, and you're starting to see the fragment getting replicated. I mean, that's this darker kind of coil here that you're seeing up at the top, and it will go all the way around, and then when it finally hits this portion, you're seeing this protein kind of fly off, and another one's coming in to create another loop or another fragment. So those are the order or the process of how this helicase does its job.
copied one section at a time. The end result is two new DNA molecules.
Okay, so that's where we'll stop with the video again. We're now about 3 minutes into it, 2.5 minutes into it, and the next portion is called transcription. And transcription we will start talking about in the next video. And after transcription, we'll talk about translation. So we're taking it in baby steps, But again, three major components that we need to look at. One was replication, which we just now finished up, and then we need to take a look at transcription, how it writes a message, and translation, how it relays the message.